This method makes it possible to manipulate and quantify how the cells organize collectively. This is important because we can model patterning in vitro and disentangle cues tightly coupled in vivo. The technique requires minimal specialist equipment, allowing it to be established in a standard biology lab, and it is quantitative, making possible the discovery of subvisual patterning events.
This method can easily be applied to any cell system where patterning could emerge, and to discover non-random patterns of differentiation, proliferation, and cell-to-cell competition. The main bottleneck with this method is the systematic optimization of micropatterning parameters for new cell types. For example, pattern size and shape, type of matrix, and cell adhesion time.
Wearing gloves, place a piece of laboratory film into the bottom of a 10 cm square petri dish. Use a 12 mm hole punch to cut hydrophobic plastic slides to create 12 mm round cover slips, and place the cover slips in a new petri dish. Use tweezers to carefully remove the protective film from the cover slips. Place the photo mask on a clean and stable surface, chrome side up, and add a 2 microliter drop of double distilled water at the position of the desired chip design. Gently press a cover slip onto the drop and place the holder on top of the plastic slides. Carefully fix this sandwich with clamps to maintain the plastic pieces in contact with the photomask. Place the assembly in an ultraviolet ozone lamp approximately 2 cm from the light source for a 10-minute illumination. At the end of the illumination period, grasp the sandwich with the photomask at the bottom and carefully